Hello everyone and welcome to today's Lab Tools webinar. I'm Catherine Lloydell, Scientific Technical Editor for The Scientist, and I will be moderating our discussion. Today our speakers Dr. Peter Kern and Dr. Wolf-Dieter Woody Fessner will be discussing the identification of novel biocatalyst candidates via high-throughput stability monitoring. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentation. To ask a question, simply click on the Ask a Question tab and type your query into the question box located on the bottom left of your screen. We'll try to address as many of these questions as we can during our Q&A session. Our webinar platform is user-friendly. You can expand the presentation window by simply clicking on the diametrically opposed arrows in the upper right-hand corner. This will maximize the display within your screen. The webinar will be archived on the Scientist website, and we will send you the link by email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our Lab Tools webinar sponsor. Nanotemper Technologies is committed to the best customer experience and enabling researchers to easily, efficiently, and accurately characterize proteins. With systems, software, and consumables for evaluating binding affinities, protein stability, and protein quality, scientists in pharmaceutical, biotech, or academia will find an optimized workflow, quality results, and responsive customer support. Our sponsor has provided us with some helpful resources related to the identification of novel biocatalyst candidates via high-throughput stability monitoring, and we have posted these in our resource list located on the left side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker. Dr. Peter Kern studied molecular biotechnology at the Technische Universität of Munich and the Tokyo Institute of Technology. She went on to do her PhD in medical microbiology at the Ludwig Maximilian University of Munich, where she studied Helicobacter pylori and its effects on human T cells on a molecular level. Peter joined Nanotemper in 2015 as an application specialist in the customer support team. For the past 2 years, she has been a product manager for the Prometheus and Tycho product lines. Dr. Kern?
Thank you very much for the introduction, Catherine. Hello, everybody. It's my pleasure today to kick off this webinar with an intro to protein stability, the Prometheus system, and nanoDSF. And I'm very much looking forward to Professor Fessner's talk following mine. Now, to start off, I want to start very basic, although I'm pretty sure most people in the audience are proficient with this topic already. But just in case somebody's in there that's not, why would you even want to look at protein stability? And I want to use the example of thermal protein stability to explain why this is useful. So you might want to understand your protein's thermal stability if you're looking for a protein variant that is suited for a high temperature application or that is suited for high temperature purification approaches. You might also want to use thermal stability as an indicator of overall stability, because this gives you a way of quickly and easily analyzing your sample to find the best optimal formulation, buffers, storage conditions, and so on, because of course you want to keep your protein stable over time. What you might also want to do is to characterize your protein structure and its structural integrity. With Prometheus you can do this on a domain level, so to see the individual protein domains and their stability and how they relate to each other. So all of these are reasons why protein stability is interesting, because of course in and of itself it's just a number that you get on your protein. Now Prometheus can help you with that because it was designed to look at protein thermal stability in a very straightforward and simple manner, but it can also do more complex things like looking at thermal unfolding and refolding kinetics, looking at the chemical stability of proteins, and also looking at aggregations. All of these are different aspects of protein stability, and it helps to have the full picture. I have examples of all of these coming up in the presentation, but first off, just to very briefly introduce you to the technology, what we do in Prometheus is we use the protein's intrinsic fluorescence to monitor the unfolding process. The reason why we can do this is that the fluorescent residues in proteins, most notably tryptophan, are typically buried in the hydrophobic core of the protein when the protein is still folded. Now, when the protein unfolds, the tryptophan residues get exposed to the buffer, and this changes their environment and thereby their fluorescence properties dramatically. So in the protein's fluorescence, we can see the unfolding happening. In Prometheus, we use either a temperature ramp to unfold the protein or a denaturant gradient for chemical unfolding. Typically, we start out with a low fluorescence signal for the folded protein and end with a higher signal for the unfolded protein, the way it's shown in this graph. It doesn't always look like this, but this is the most typical profile that we see. And of course, the halfway point of this process is what we call the TM, the melting point, or the CM in case of chemical denaturation. That's the point at which half of your protein is unfolded. And this can be used as a parameter to compare protein stability across samples, across proteins, across buffers, and so on. Next, I'd like to present some examples of applications for Prometheus. We'll of course hear a lot more concrete examples later, but just to give you an overview of things that have been done and can be done. My first one is kind of basic, but it really shows the principle and what you can do with the system. It's an esterase that was analyzed by a research group in Jülich, Professor Karl-Erich Jäger and his group, where they looked at this esterase in different detergents. A lot of the time you need detergents to stabilize your protein, but not all of them do that job equally well, so you might want to analyze which one does it best. You can see on the left side the unfolding profiles of this protein in the different buffer conditions. It's all different detergents that we use, including a non-detergent buffer, which is the darkest of the lines that you can see. Now you can also see that most of these profiles look very similar to the generic one I showed you before, but two of them don't. The red one and the green one just show a flat line. These are two detergents that apparently don't stabilize the protein at all, so it's already folded at the beginning of the experiment, which is why we don't see any unfolding process happening anymore over the course of the temperature ramp. For the others, we do. We start with a low signal and end with a high signal, but you can see that the profiles are shifted there. Excuse me, because the different detergents influence the stability, so we get a shift towards lower temperatures for most of them, meaning they destabilize the protein. Another way of looking at this data is shown in the bottom panel. This is not looking at the fluorescence data directly, but instead looking at the first derivative of that. And the only reason why you'd want to do that is that it's easier to analyze visually, because what you get is peaks instead of these S-shaped curves, and those are always easier to identify. But they show you the exact same thing. You can see the buffer condition is the rightmost one, and then we have a shift to the left to lower temperatures for the destabilizing detergents. What's especially interesting about this example is that the group also compared this data to enzymatic assays that they did with the same esterase, where they did this enzymatic assay at different temperatures to see at which temperature the function of the protein gets lost. And while the absolute numbers that they got were different, they were able to get the same result in principle, so seeing that those detergents that unfold the protein also, of course, completely preclude any enzymatic activity. The buffer condition that is the most stabilizing to the protein, and the DDM condition that show a similar thermal stability in the Prometheus measurements also show a similar enzymatic TM, and the last detergent is in the middle. So with this, you can get the information you can get from the Prometheus run is which condition is the best, where is my protein still active. The same type of information you can get from the enzymatic assay, but running this on Prometheus might be a lot faster. My next example also shows an enzyme. This is a lipase, a very different type of enzyme, of course, and its thermostability in methanol. And this is, of course, interesting because not very many systems can use ethanol buffers. A lot of them use, for example, plastic sample containers that don't allow this type of harsh condition. But in Prometheus, we use glass capillaries as sample containers, and they are perfectly happy with methanol. For this application, of course, it's crucial to be able to do this because this lipase is a candidate to be used in biodiesel production. where you need enzymes that can withstand all of these types of conditions. So this group from Technion in Israel was looking at this protein. You can see the unfolding profiles here for also a buffer condition, the black peak, and then with increasing methanol condition, we get more and more shifted to lower temperatures. So the higher the methanol concentration, the less stable the protein. Not very surprising, of course, but the interesting part, of course, is when they mutated the protein and tried the mutant, because of course they were looking to stabilize it to be able to withstand these conditions better, and they found that the mutant was indeed more stable in all conditions tested. So a very straightforward way of looking for that variant or that protein that is best suited for what you want to do with it. In my next example, I want to take a very different approach to looking at protein stability. This is a completely different type of protein. This is an antibody, but it doesn't really matter, so Prometheus will happily analyze any type of protein for you. Here we looked at antibody and their unfolding reversibility. We have a dedicated software for doing this type of experiment. It's called PR time control software, and it can run different types of temperature profiles, not just your average linear ramp that you typically run, but what you can see here is a incremental temperature cycling experiment. So in this first graph, you can see a dotted line in the background. This is the temperature profile. So you can see that we started with a very low temperature, then increased the temperature a little bit and came back down to base temperature. In the next cycle, we increased it a little more than before and then again came back down to base temperature. And we keep doing this, increasing with every cycle until we reach pretty high temperatures at the end. And what you can see in the blue graph, this is the fluorescence signal of the antibody. You can see that for the first few cycles, nothing really happens, right? The line is pretty flat. And then finally, at some point, you see a small bump for one of the cycles, and then the bump gets bigger and bigger with every cycle. But the signal always comes back to the baseline for a few cycles, meaning that the protein is unfolding with the increased temperature, but as the temperature goes back down, it folds back. At some point, this doesn't happen very well anymore. We don't get back to baseline anymore. Instead, some of the protein at least stays unfolded. When we compare this to a different antibody, we see a very different picture. So here now on the right in this red profile, this is a second antibody that shows completely irreversible unfolding from the start. You can see the signal increasing as soon as the first temperature cycle is run, and it never comes back down to base. So very interesting to see this comparison side by side here, but what gives this even more weight is when you look at the aggregation data in parallel. So Prometheus collects these data, as I said, in parallel on the exact same sample during the same run, so there is no extra time needed and no extra sample. And you can see the results in the bottom panels here. So if we look again at the first antibody, the blue one on the left, you see the unfolding happening in the top part and then the aggregation happening in the bottom part. And again, you can see a flat line for quite a while until after the third peak when there's finally an increase, meaning that there is actually aggregation happening in the sample. We see that this happens as soon as the unfolding process also stops being reversible. This is a common correlation that aggregation happens as soon as there is irreversible, or aggregation is the cause of the unfolding being irreversible actually, depending on the protein. So this correlation is not surprising, but it's really important to know, of course. For the second antibody, we see the same correlation, but it happens a lot earlier because, as I said, this one doesn't fold back from the beginning. So this can be really a good method of looking at your protein stability from various perspectives. And again, for example, looking for that protein that can withstand some type of industrial process that you have or some type of temperature conditions that it'll be subjected to during its lifetime. My final example is again very different perspective. So this is about chemical denaturation. Of course, chemical unfolding experiments are not new. They're very much established. You use them to understand the free energy of folding for your protein, and you do this by subjecting your protein to different concentrations of a denaturant. Now this in and of itself is very straightforward, but you can do this on a kind of a meta level. This is what we did here, because it's been postulated that if you measure delta G for different concentrations of your protein in addition to different concentrations of your denaturant, then this may help to predict aggregation propensity of the protein, meaning the likelihood of the sample to aggregate over time. And in a study that we did with Boehringer Ingelheim, we were able to show exactly that. So here you can see what we did there. This was actually a whole lot of measurements. It's all condensed down to a few graphs here. We measured, or they measured, delta G for an antibody at 4 different concentrations in 5 different formulations. The different graphs here are for the 5 different formulations, F1 to F5. And we are looking for a trend in delta G. Now, we saw on the slide before that any increase in delta G with protein concentration means the protein is aggregating in the native state. Any decrease in delta G over protein concentration means your protein is aggregating in the denatured state, while any type of horizontal line means that your protein is aggregating a lot less. So this is typically the condition you're looking for, and our experiments identified formulation number 5 to be the best one because you have the least trend here. It pretty much varies around 0, while for the other ones we all see this decrease in delta G, meaning that there is aggregation happening from the denatured state. Now we of course confirmed this. Just predicting this doesn't help you much, but Boehringer actually took this antibody, stored it for a year or a year and a half, and then looked at its aggregation state via turbidity measurements and monomeric content and stuff like that, and they identified the same formulation to be the best one. In addition to that, we also did thermal stability measurements. We also looked at the aggregation behavior directly via Prometheus, and they all agreed on formulation number 5. So if this indeed works out for your protein, then you can skip a lot of time and a lot of tedious steps because you don't have to wait for the whole year. Instead, you can predict aggregation propensity early on and weed out those conditions that don't look promising. All right, so this was a lot of different ways of looking at protein stability. Now I'd like to explain a little bit more about how this is done inside of Prometheus. Firstly, I've mentioned the capillary format before. Here there are shown the capillaries on the thermal element. Running an experiment is really simple. So you load your sample by dipping the capillary into the liquid. It'll automatically pull up the liquid by capillary force, and then you put your capillary on the tray and you close the instrument and press start. So it's really, really simple. The capillaries hold 10 microliter each, so that's really not a lot of sample. And then when they sit on the thermal element, they can be heated anywhere between 15 and 95 degrees. Once you start the instrument, it'll start scanning across those capillaries and reading the fluorescence all the while, meaning that with each scan across the capillary tray, we get a data point, and this happens every 3 seconds. So that's a lot of data points. And then, of course, what we get out of this is the fluorescence signal. And we collect our fluorescence signal actually only at 2 wavelengths, which is at 330 nanometer and at 350 nanometer. And I want to explain briefly why we do it like this. So in this image, we see the fluorescence emission spectra of a protein at its folded state in gray, at its unfolded state in blue, and at a transition state in red. And you can see that with the unfolding process, typically you get a reduction in emission intensity, so the peak gets flatter, but also you get a shift of the peak from 330 nanometers to 350 nanometers. So this is what we see in most proteins, and actually we don't need to have this full spectrum to understand what is happening. By looking at 330 nanometer and 350 nanometer, we get all of the information that we need because we can see both the shift and the reduction in fluorescence signal. And by the way, all we need is a change in fluorescence anyway. So this is what we do. We collect that over our temperature ramp or over our chemical unfolding experiment, whatever it is that we're running. And we typically get something that looks like here. In the center panel, we have, of course, both of these wavelengths detecting the fluorescence. We typically get a decrease in fluorescence over temperature anyways, because that's just how fluorescence behaves. But then what we typically look at is not this, but instead the ratio of the fluorescence at 350 divided by the fluorescence at 330 nanometers. This gives us the characteristic S-shaped profiles that I've showed you before. We do this because it's a little bit more robust against effects like air bubbles and things in the sample, and it's easier to interpret most of the time. And in addition to that, you can look at the first derivative shown in blue here. This is what I showed you before too. If you're more into peaks than into transitions that you might be used to from other technologies, for example, then this is perfectly fine as well. But that's all you need to know about the fluorescence detection in Prometheus. I want to also mention how we detect aggregation, because this is also a really cool and really simple concept. So it's shown here on the right how we do it. We have our capillary containing the sample, and we shine some light through the capillary. The light then gets reflected back because the ground that the capillary sits on acts like a mirror. So the light gets reflected back, and more of it gets reflected back if there is no aggregation. But if there is aggregation in the sample, some of the light gets scattered, and less of it gets back reflected into the detector. So it's a really simple type of measurement, but that also means we can do it really, really quickly, and we can do it at the same speed as we do our fluorescence measurements. On the left side is shown what typically happens in the sample and what that signal typically looks like. So we start with soluble protein. It starts unfolding at some point. This point is what we call the onset of aggregation. That's also the parameter that the instrument outputs for you and the parameter that you use to compare. And then at some point, the protein is fully aggregated and we get a plateau in our curve. This is an optional upgrade for the Prometheus. You don't have to get it if you don't want it. And now I want to sum all of this up a little bit by explaining why you might want to use, choose Prometheus if you're looking at protein stability. I think I've mentioned most of this already, but just to sum up, Prometheus helps you because it means you can trust your results. It gives you such a high data point density, such a high resolution and precision that you can be sure that you're not missing anything, and you can be sure that what you're seeing is actually real. Also, you can use it for pretty much any project and any target protein, because it doesn't care what protein you're putting into it. You can use any type at pretty much any concentrations, from really low to really high. You only need 10 microliters of sample, and you can use any buffer, as I've shown, detergents, solvents, anything. And also, it's flexible and fast. You can change your ramp to a faster ramp if you don't have the time. You can choose to look only at the temperature window that's interesting for you. So you really have all of the control that you might want. And then as a last thing, I only want to mention again that while Prometheus can help you look at protein stability, we also have the Tycho product line in our portfolio to look at protein quality, and the Monolith product line to look at binding affinity. And if you're interested in any of those, of course, I'd be happy to answer any questions on that or anything else after the webinar. For now, I'm at the end of my presentation. I'm looking forward to Professor Fessner's, and with that, I'd like to thank everybody for your attention.
Thank you, Dr. Kern. As a reminder to our audience, you may ask questions at any time using the question box. Our speakers will have the opportunity to address them during our Q&A session at the end of the presentation. Our next speaker is Dr. Woody Fessner. Dr. Fessner holds a chair in organic chemistry at the Technische Universität Darmstadt, Germany. He obtained his PhD in 1986 and then worked as a postdoctoral fellow with George Whiteside at Harvard University and George Olah at USC, LA. Before moving to his current position in 1998, he was professor at RWTH Aachen University. His research interests are in the area of biocatalysis for applications in organic synthesis, starting with a 1992 landmark development of in vitro multi-step artificial metabolisms, now known as systems biocatalysis or synthetic biology, with particular emphasis on the discovery and development of novel enzymes for stereoselective carbon-carbon bond-forming reactions and the synthesis of complex oligosaccharides. His group is also interested in the application of directed evolution technologies for the development of biocatalytic protein modules with functions specifically tailored for cascade applications. Currently, he is coordinating the Horizon 2020 project, Carbozymes. Dr. Fessner?
Thanks, Catherine, for the introduction, and thanks, Bea, of course, for the excellent intro into technology. Welcome, everybody in the audience, and thank you for your interest in our little study in protein stability using the Prometheus instrument. Now, this, as you just heard, is a presentation that collects results from a collaborative research project that is funded by the EU. It contains about 14 partners in 6 countries and is operating since 2015. Targeting applications of enzymes for carbon bond formation. In this respect, we were interested in studying these enzymes, particularly for stability, for process conditions. Now, the presentation, this is the overview. I will very briefly introduce why we are choosing enzyme stability as a target property to investigate. I will tell you about solvent stability of enzymes. Then we deal with a third study directed at library studies, so it's about throughput. And the last topic will be about discovery of robust enzymes from nature that have not been characterized before. Now, this is only a very brief recapture of why biocatalysis is interesting. It's regarded as a key enabling technology for future industrial processes that are more sustainable and in particular less energy demanding, and therefore will hopefully leave a lower environmental footprint on our planet. In general, this is something that is interesting not only for fine but also for large bulk chemical synthesis. Now, industrial applications, of course, they require enzymes that have particularly suitable properties for industrial use. First of all, they have to withstand many stress factors. You have to remember that most of the substrates that organic chemists are interested in have a rather low aqueous solubility. So usually to reach higher substrate and product concentrations to make processes more economical, you will have to utilize a number of additives like co-solvents or maybe even surfactants. You might need to change the pH from neutral, or you might need elevated temperatures. That is, although I mentioned that these processes should be less energy consuming, elevated temperatures are useful, and therefore thermodynamic protein stability is an asset. So proteins that are less tend to unfold under process conditions will last longer, and therefore it will be more economical. It also helps in protein purification if needed, because by simple heat treatment you can get rid of most of the host proteins, and only the interesting proteins will remain in solution for your catalysis. At higher temperatures, of course, processes can be run faster, reactions are faster, catalysis is faster, and therefore you can gain a lot of profit from higher reaction rates. And of course, long-term use of proteins and recycling in addition is something that improves much the cost efficiency. Now, the first study I would like to deal with is we studied the response of proteins, and particularly different proteins, to organic co-solvents. And in this slide, you see that as an exemplary and relatively sensitive enzyme, which is hexokinase from rabbit muscle. It's a commercial preparation that we used that can be utilized for several purposes, and it's also used for assay purposes. Now, the thermal unfolding of that gives a very clear curve. This is the green one if the enzyme is treated to higher temperatures in plain buffer. If you change the situation and by adding acetonitrile, for example, this is the blue curves with very few addition of acetonitrile, 1%, 2%, up to 10%, you notice that the unfolding starts at very low temperatures already. And the same is true if you utilize tetrahydrofurane, with both of which are solvents that can be used to solubilize organic materials in aqueous, under aqueous conditions. So this is something where you very soon reach the limits of protein stability under reaction conditions, And although this is not the best enzyme to utilize for technical purposes, this is a certainly exemplary stage that shows that, first of all, you observe under those conditions, and by adding co-solvents, you obtain clear melting curves. So it's very easy to interpret those. You also notice that there's a proportional response to solvent fraction. This is something that Bears already introduced briefly with the study of this lipase in methanol. But it seems to be that this is exactly proportional. And you also notice that the data quality is rather good. So the curves are well behaved, even down to room temperature at 25 degrees. So you can directly start monitoring that from ambient temperatures. This is just a single enzyme. This is hexokinase. But we've studied a number of these enzymes, and particularly ones. This is the hexokinase sample and at the bottom. Enzymes for carboligation, like transketolase from Geobacillus or deoxyribose aldolase from Thermotoga, a hyperthermophilic enzyme apparently. But all of these, and you notice that on the right-hand side you see the different solvents that we tested, all of these respond in a proportional fashion to increasing level of co-solvent. We've tested, as you notice, up to 50% with those that are more tolerant. But you notice that DMSO and DMF, the green curves, are relatively well tolerated. Alcohols, like the blue symbols, they are kind of mid-range. And they are the bad solvents, the ones that are purple or red with different symbols. Those are the harsh solvents that are good for solubilizing organic material, but these apparently are the ones that are most complicated to use with enzymes. So this is a set of 3 different enzymes. There's another set, a second set that we investigated. Some enzymes that are interesting for carboligation, like FSA and another DERA, the one from E. coli in this case. All enzymes from hyperthermophilic down to mesophilic, like this alcohol dehydrogenase from lactobacillus, they all give similar proportional response. And although we have utilized very different types of enzymes, all show very similar and almost linear behavior. So this is something where apparently it is sufficient to just monitor a few data points to get a good picture for extrapolation of possible reaction conditions for preparative applications. So this is a summary slide. We've called that a kind of a response factor to the different solvent types. And you may notice that the left one, DMSO, is one that has very little effect on enzyme stability. And of course, this is an empirical observation that is known from many literature studies, that DMSO is a solvent that can be utilized up to very high concentrations. We've actually noted that very early on studies in carboligation 30 years ago. And DMF is something that sometimes is well tolerated, similar to acetone, here called propanone, or different alcohols, primary alcohols like methanol and ethanol. Secondary alcohol 2-propanol is a bit less well tolerated. And acetonitrile and THF, I just mentioned, give very strong destabilization effects on proteins. So this is something that is certainly difficult to treat theoretically because we do not see a direct correlation with physicochemical properties known for these solvents, such as log P, etc. There's a tendency, but no really clear correlation that can be drawn from that. We've also looked at the structures of these enzymes and tried to correlate some of these properties. So they are dimer, decamer, and tetrameric and 1 monomeric enzymes. All of them can have very different TM values, of course, depending on the organism where they came from and depending on the evolutionary pressure on the structural side. But there's no evident correlation of the structure except for the fact that maybe transketolase can unfold because the active sites are jointly formed by both subunits together. They're also the cofactors bound at the same site. So once these dissociate, the protein becomes more vulnerable to degradation. The same is true maybe for hexokinase, because the monomer you see has two different protein portions that have a conformational mobility. By substrate binding, the protein is conformationally tightened, so the active site is closed for conversion. For the CRED is a tetra, it's despite that quite flexible protein, so there's no direct evidence for that. If you look more closely, and this is from a study from Dick et al. That was recently published in Scientific Reports 2016, they've investigated two different DARAs. One is the DARA from E. coli on the, in the center picture. This is an enzyme from a mesophilic organism. And on the right-hand side, you see the enzyme from Thermotoga maritima, apparently an extremely stable enzyme. And you can clearly see from the structures of these proteins, and there the authors have investigated the intra and inter subunit forces, so stabilizing forces that stabilize the subunit by themselves, but also the forces that stabilize the interactions of the subunits at the interface. The mesophilic enzyme shows much less attractive forces than the other one. Of course, this is to be expected, but if you look at the left-hand graphic that I added to this, you notice that both of these enzymes, so the red bars are for the E. coli, the mesophilic enzyme, and the orange ones for the hyperthermophilic enzyme, both show almost exactly the same response to the different solvents. So apparently that tells us that it is something that the dissociation of the subunits and the unfolding seem to correlate quite well. Both seem to happen simultaneously. At least for that particular enzyme type, this is quite evident. Now, let's give you a few conclusions from the first section. We believe that the nanoDSF technology with this instrument is highly efficient for quick and for reliable screening of reaction parameters. Dilution series are easily set up. You can vary a few values just only for linear correlations. Extrapolation will give you very likely correct values for extra data points. You can, of course, simultaneously screen with 48 samples several solvents at the same time. You can also screen for other factors at the same time. For example, substrate, product concentration, additives like salt surfactants or buffers, pH variations. Everything in principle can be done in just a few runs, and you are quite likely to identify the correct or appropriate reaction conditions that match best your desired process. Now, this is the second set of examples. It's about protein engineering. We are interested in utilizing directed evolution. That means we are utilizing mutagenesis, saturation, mutagenesis, site saturation, and that gives relatively large libraries of proteins. And the problem with that type of research is that mutagenesis usually causes defects in the proteins. This causes destabilization of the protein fold, and therefore in enzyme engineering projects, stability typically is at risk. And if you risk the stability, then you cannot go very many cycles, so it's important to know about the stability of the protein at every stage. If you talk about libraries, if you talk about larger numbers of proteins that have to be investigated, this means you need high throughput preparations. There are many steps involved. Cell growth, you need centrifugation for separation. Then you have cell lysis, again, centrifugation step. And usually for most of the techniques that are appropriate for monitoring protein stability or protein unfolding, you need purified protein. So for example, affinity purification with a tag requires binding, washing, elution steps, and all of these steps, of course, add up. These are tedious, they produce errors, and of course, they are costly. In addition, data variation is something that very often occurs. Even if you substitute manual operation by automatic operations, very often you have error factors and data variation that cannot be obviated. So that leads to the point that a direct TM measurement from lysates, from unpurified proteins, is highly desirable. Of course, direct TM measurements depend on sample purity, because contaminating proteins will also give these melting curves, and the background of these curves, and there are loads of different proteins in the host cell, they will give you a background noise that might completely overshadow the signal that you're interested in. That means you have to optimize a few factors. The overexpression rate should be relatively high to supersede the host proteins. And the cell lysis method is also something that is to be considered, because if you introduce reagents, for example, this will definitely also have an impact on the protein stability, on the data quality that you will receive. Now, we investigated a number of optional methods for cell lysis, and of course, if you go for libraries, then you have arrayed proteins in plates, and of course, mechanical methods cannot be used under those conditions. So sonication, parallel sonication, or treatment with enzymes like lysozyme, nuclease, or using detergent or freeze-thaw and osmosis techniques are quite common. And we found that our preferred method, which we call hypotonic extraction, is quite satisfactory for that purpose. So frozen cells that directly come from the overexpression, so from deep wells, you centrifuge, and the frozen cells then are suspended in appropriate hypotonic solution, hypotonic buffer solution at room temperature. Then the cells will be cracked and proteins released. And if you utilize, for example, 10 millimolar tris at pH 7.5, this is something where most of our proteins are stable and active, will give you the appropriate protein solution. We did not test other systems, but I'm sure other buffer and solvent conditions can be utilized. So this is then from a study where we've tested enzymes from a deep well growth with different conditions of solubilization. The bugbuster is one of the commercial reagents that is sold for cell disintegration, and this is now confronted with a hypotonic extraction technology that I just explained. The detergent treatment, as you see from the left-hand page gel, this gives a higher protein yield. So the quantity of protein that is extracted from the cells is higher in total, but you see a lot of background protein from the host cells. And the protein, the desired protein, the one with the green arrow, is maybe only about 30 to 40% of the total protein content. For the hypotonic extraction, you see much less of the protein background from the host cell. So I judge that maybe about 50, even 70% of the protein that you extract from the cell is the desired one, so you have less background. So the protein is in a higher purity state, although the quantity is less, the purity certainly will have an impact on the data quality. We've tried also to compare the data that are generated from these two different procedures. Now, from the hypotonic extraction procedure on the right-hand side, the blue bars, on average, we see a TM of about 63 degree with a very good reproducibility. And on the left-hand side, you see the TM values determined after bugbuster treatment of the cells for lysis. So very clearly, there's a strong destabilization effect by the detergent. In this particular case, it's more than 8 degrees reduced value. So definitely, if you're utilizing any kind of chemical treatment of your cells, overexpression cells, you definitely will have an impact on the data that you generate from the measurements. And the true values, well, the question is whether this is unaffected at all, are from the hypertonic extraction, where we have the least concentration of additives at all. So we've investigated that in the context of this carbozymes project for developing a software, well, not we have developed the software. This is from the company, the SME company Bioproduct, another partner in the consortium, and they were interested in experimental data to support the software development, particularly from mutagenesis studies where only little modifications in the protein have been performed. Now, just a very brief outline of a, well, directed at cost efficiency strategy. So we utilized a particular enzyme, this DERA scaffold, because there's a high resolution X-ray structure available, identified together with the 3DM analysis of Bioproduct 6 positions where likely modifications will cause larger effects. We've done the saturation mutagenesis, those 6 sites, measured the TM in 96-well format, sequenced selected variants, and repeated that at a different DERA scaffold to investigate how these data can be transferred to a different protein scaffold. So all in all, you see that we have to run 2 by 6 by 96. That means 1,152 total data sets, and a part of these we have even done in triplicate to make sure of the data quality. So this is definitely something that profits from throughput. And this is just as an example from one of these runs, half of a plate, 48 samples altogether. This demonstrates that the background, there's a negative control, which is the black curve, does not give any signal, but all of the wells that we investigated gave a rather good signal. So all of these can be interpreted quite well. Clear curves, clear melting points, and almost no failures from this treatment. If there was a failure, then very likely it's a failure of expression for whatever reason. And of course, data were highly reproducible from the triplicate measurements. So the next slide is a summary just for one entire plate. This shows, first of all, that the entire plate from 2 runs generates the corresponding data that are sought for. There are 2 negatives. These were the negative controls. There's no gene and therefore no protein. And the wild type is, in this case, on the right and left-hand side. So this tells us that all of the mutants introduce some destabilization. And we've sequenced a number of these to get at least 15 different identified of these mutants. And you see also the data variation. This is the red bars. They show the statistical spread of the data. So all in all, this gives quite a good data situation. I forgot maybe to mention that you see the lowest spots, the lowest data marked with a P. This is from identified from this slide here. All of the red curves do contain the proline, and the proline is known in many other studies that this is quite destabilizing in several occasions. That was directly expected from this investigation. We also tried to cross-validate our data with other methods, and particularly there's one method available in literature that is called thermal shift assay. It's an established method that is using an environmental sensitive dye, which is a Cypro orange commercial dye that is added if the protein unfolds. This fluorescent dye varies the fluorescence, and this indicates then the unfolding event. So this can be measured quite easily, similar to the instrument under discussion here. However, we do see a discrepancy in the data. First of all, we do see that the blue bars from nanoDSF are always higher than the orange bars that come from the Cypro measurement. And we do see, and let's just focus on the middle one, the 55X library, for isoleucine replacement in the position X, the data are almost identical. So the orange is only slightly lower, but for the arginine replacement and for the tryptophan replacement, this is highlighted here, you see a very strong discrepancy. And apparently, and this is a conclusion from several of these investigations and also utilizing other mutants for analysis, apparently there's a sequence-dependent destabilization effect. Apparently the Cypro dye causes a variable TM deviation depending on the sequence. And that is clear if you look at the structure of the Cypro dye on the upper right-hand side. It's a Zwitter ionic dye that also contains a large aromatic portion. So pi complexation certainly is an issue. And the effect of association with a protein upon unfolding is less so for hydrophobic residues, but is quite large for polar, due to the Zwitter ionic portion, and aromatic amino acids. So particularly for tryptophans and phenylalanines, there's quite a strong effect that we've found. And this clearly influences the unfolding equilibrium, and the data definitely are not correct that you measure with this method. Now the conclusion from the second set is now that the method is quite effective for high throughput determination of protein melting points. You don't need tedious protein purification if you have sufficiently pure protein from sample preparation by hypertonic extraction, which is hypertonic lysis for frozen cell pellets. You get true values because there are no additives involved, and the manual throughput that we have found in our lab is either 288 samples or maybe even up more than 300, depending on the person who is running that. And definitely this can be accelerated by higher heating rates or faster ramping, maybe even with automation. Now the third, and this is just a small story, this is about an enzyme, DERA, which we've seen quite a lot. This enzyme is crucial for the synthesis of the statin compound class. It's a drug class for cholesterol treatment, and these enzymes are deactivated under process conditions. They are useful for stereochemical integrity of the product as needed, as you see on the right-hand side of the chemical reaction here. The enzyme repetitively does the carboligation step, but the enzyme under the process conditions dies and cannot be regenerated. So we were looking for metagenomic orthologs of these known enzymes, and of course this is something where you need panels of maximum diversity, phylogenetic variation. This is something where we profited again from one of the SME collaborators in this consortium, the company Prosomics Limited from UK, and they have assembled panels of these DERAs in 96-well format. And we've measured them. So this is, first of all, just to demonstrate that the protein that they provided to us, cell-free extracts contain a lot of these proteins because of good expression rates. So again, the green arrow highlights the protein in question that is interesting to us. And most of these proteins are quite well expressed, maybe except of number 9, which is the right-hand, on the left-hand side, the rightmost, or again on the rightmost side of the right-hand side, number 17 and 18. So these proteins are not well expressed, but still we've investigated everything to try to get TM data for these proteins. I do have to introduce you to a study by Wedde et al. That was recently published in ChemBiochem, where they've tried to measure how much protein you need against background. So they've done a titration experiment where they started from the red bottom curve, which contains only crude extract, and then added small portions of a purified cyclohexanone monoxygenase, which in pure form gives the top blue curve. So this literature study shows that it needs about 30 to 40% purity of the enzyme should the measurement be clear and sufficiently clear to give a good reading. And this CHMO actually is a special case because it does contain a large number of tryptophans and therefore provides for very high sensitivity. But I can assure you that even proteins that only have a single tryptophan in our hands gave good reading if it is about 40% of the total protein investigated. And that is shown here in some exemplary readings. So the red lines are the ones that are for those enzymes with good signal quality because of the relatively high expression rate. So proteins number 1 to 5 are quite easily identified against the background, while the blue curves indicate those proteins that did not contain a tryptophan residue. So for example, there are 12 and 15 from these, and that is something that only gave host protein background. And of course, this is something you also notice the background is not uniform. Sometimes you have different smaller peaks from particularly in the center region from host proteins that have possibly different expression rates. So it's not easy to subtract just the background for noise correction. Now from this total analysis, the first pass of this 96-well plate gave 53 immediate TM ratings. So this is about 55% of success. And for a second rescreen of some of the low-quality scans, we got 3 more TMs. So this is a total hits of 56 readings out of 96 samples, which is about 58%. Of course, you realize that some of the proteins might not contain a tryptophan residue, and from sequence data, we gathered that 24%, which is 1/4 of the total, did lack a tryptophan residue. So actually, the yield, if you like, is actually quite higher. 3 proteins that did not contain tryptophans still gave nice melting curves. They contained a relatively high number of tyrosine residues that were appropriate also to give good readings. And 15 samples altogether expressed weakly, and this is maybe to be called false negative, so 20% dropout were not observed, although a tryptophan could have been in place to be monitored. These are the ones that, because of low expression, did not give readings. Now you notice that on this left-hand side, where we have the corresponding tree in circular format to show the relationship of the proteins, together with 5 known reference enzymes highlighted in green numbers, we get very good readings, and these are highlighted in the sun representation in different colors. So the blue colors are enzymes that have rather low TMs, and the red ones are the hot ones with the TM that even have more than 100 degrees TM. So this is something that can be measured only with closed capillaries, obviously, but that is truly easy to monitor with this particular instrument. So that means better than 40% of the enzymes were discovered to have high to very high TM. So these are enzymes with very likely robust nature that might be of potential interest for industry. And of course, we tested then for activity, and this is now this graphic over here, where for the entire plate, we've collected activity data plus color coding for the thermophilic enzymes. And you notice that in the center of the graphic, there are 2 enzymes highlighted in red that give quite good activity and that are highly thermostable. And there are more enzymes that have very high activity and still good stability data from this analysis. So this is something that is going on, of course, and I will stop with that. Just for the conclusions, we believe that nanoDSF is quite useful discovery tool to find thermostable biocatalysts from protein libraries, particularly from metagenomic libraries where you do not know where the proteins come from, where you do not have clues whether it comes from a mesophilic or from some thermophilic source, and even from lyophilized CFE powder of these enzymes, you might be able to directly measure without any purification need. And of course, if you get positive hits, they are direct candidates for industrial use. And as I've shown you, of course, this has to be correlated with activity. Clearly, the technology does have limitations. It does require proteins with at least one tryptophan. Most proteins do contain tryptophans, but for the DERA study, you noticed 25% in our array did not contain tryptophans. Or the protein should contain many tyrosine residues so that it is possible to retrieve some collective data for those. And of course, again, low expressing proteins will be missed, but of course, if you're interested in those, they can be purified if you tag them, and then of course you might be able to gather more of the data. So this is from my side. Thank you for attention. This study is collected in a manuscript which we have submitted for publication in FEB's journal. The manuscript is currently under revision, and I hope it will be published soon this year. Thank you again for your attention, and I'm happy to collect questions. Sorry about that.
Thank you, Dr. Fessner. The audience has submitted several questions, so let's get to them. And here's one for you, Dr. Fessner. What is the timescale of solvent effects on enzymes, and how does activity change with time after transfer from aqueous to co-solvent regimes?
Well, of course, we did not investigate the timescale. If you heat the enzyme with a different ramping rate, then of course you obtain an unfolding. We've shown that this is quite independent of the heating rate. Very likely the data are quite close to what you obtain with a decent rate of 1 degree per minute. However, we've not performed any study of refolding, so this is something we cannot tell. And okay, activity, of course, activity we did not focus on in this study. And of course, this is a separate issue that has to be investigated separately. Right now, we're only interested in the thermal unfolding, which is a property of a protein that usually is associated also with stability against kinetic issues. So, for example, also chemical stability, although it is not directly related.
Okay, thank you, Dr. Fessner. Here's another one for you. Do you observe multiple peaks for the unfolding of an enzyme with multiple subdomains? And if so, how do you interpret the melting curves?
Okay, that's a good and interesting question. And particularly for the hexokinase that I've shown, we've utilized for that study that I've shown, we've utilized a protein from commercial sources that did contain some glucose. And glucose induces the protein to fold around the substrate. Substrate is bound at rather low Km values. So the protein becomes more compact and therefore shows only a single melting point. However, if you investigate hexokinase, which is also a 2-subdomain protein, without the presence of glucose, which we did in a separate study that I did not show here, you do observe that there are 2 maxima. This is known for that particular enzyme and has been shown with other techniques as well. And I interpret that, of course, that the subdomains might be might have a different stability internally, so that potentially upon unfolding, they unfold individually, and you obtain a first and then a second peak. And of course, because these are quite likely to be rather close, you do not really have completely separate peaks, but you see shoulders very often. Does that answer the question?
Yeah, that's a great answer. Thank you for that, Dr. Fessner. Dr. Kern, here's a question for you. Do you have any software options available to run the Prometheus system?
Yeah, so we have different software packages for different types of experiments that you might want to run. We have a software for running regular thermal ramping. This is the kind of experiment that Dr. Fessner was showing. It's called PR Therm Control software. Then we have PR Chem Control for running chemical stability experiments. And we have PR Time Control for running these unusual things like isothermal measurements, temperature cycling, and so on. We are also very close to launching a dedicated analysis software, which can then give more in-depth view of in-depth interpretation of your results.
Okay, thank you, Dr. Kern. Here's another one for you, Dr. Kern. I'm in a screening group, and are there options to automate the workflow process on Prometheus?
Yes, there are. So I didn't mention this, but I did show an image somewhere on a corner of my slides. You might have seen that. We do offer automation for Prometheus, which consists, it's called the robotic autosam, that consists of a robotic arm that does all your sample loading. So you just have to pop in your sample plates, and then you can go enjoy your weekend. And judging from what Dr. Fessner was saying, with the amount of data that they have collected in their group, they might have they might like to have that. I imagine being the person screening the 288 samples per day. So for those kinds of throughput needs, of course, we do have a solution.
Okay, great. Is there a protein concentration range that is required or suggested to be used to test on Prometheus?
Yeah, so we have a concentration range that we know works for all of those proteins that we've tested. We start at low concentration, somewhere around 5 microgram per milliliter, and we can go up to as high as 250 milligram per milliliter. Of course, it will depend on the protein. As we saw, proteins without tryptophans give a lower signal, so sometimes you might need higher concentrations of those. But really, we've used all kinds of samples from early low-scale expression up to formulation and late-stage developments of biotherapeutics. So we have really, really high concentrations, and we can do all of them. So we're very happy that we're that flexible.
Okay,
I might add from my side that for our routine applications, we utilize solutions that contain about 1 milligram of protein per milliliter. So that relates to the quantity of 1 microgram of protein per measurement, which is rather low in demand.
Okay, great. Thank you, Dr. Fessner. I think this one is for you as well, Dr. Kern. Do you see a reverse shift in fluorescence value at 350 nanometers for some proteins?
Yes, we do. So the profiles that I showed all kind of looked similar. I didn't show any of the unusual ones, but we can see pretty much any type of shape. It can be the other, I call it the other direction. So instead of having an S shape, you have a reverse S shape. If there are multiple domains, since this question came up earlier, if you have multiple domains in a protein that unfold more or less independently, like you have an antibody, for example, you will see several steps in your unfolding profile. So we've seen all kinds of shapes. The ones I showed are the most typical, though. So, but anything can happen.
Okay, thank you. Dr. Fessner, why should I measure stability for engineered proteins?
Because the next step might compromise the outcome. If you start with a protein that has relatively critical stability from the outset, and some of the proteins that we know and that are interesting for engineering, like the cyclohexanone monoxygenase that I've shown from the published study, those are enzymes where apparently no thermostable orthologs exist so far from nature. And therefore, if you do a protein engineering on a protein that does already have a limited stability at room temperature, you will miss out a lot of possible answers to the original question of improved potential. Now, the first generation might yield to enzymes that have a better property, desired property, but also very likely will lose some of the stability. If you're interested in a further cycle of optimization, then you will start from a protein that has intrinsically lower stability than from the start, and this will quite likely get close to the threshold of stability, so you will miss out many of mutations that have two detrimental, if I may call that, modifications in the protein that compromise the fold of the protein themselves. So you lose out a lot of answers of the engineering question. It's better to have something, of course, at the outset that is stable, and then you don't have to measure. You should, as I said, but particularly for those situations where you start from a compromised situation where the protein does have limited stability and you have no alternatives, then it's very important to monitor stability.
That's great. Thank you, Dr. Fessner. And here's another question for you. Aren't thermostable enzymes more rigid than mesophilic ones and thus less interesting as catalysts?
Yeah, that's a good question. We know that many enzymes that are more thermostable, they do unfold and they are more mobile only at higher temperature. For catalysis, you need, of course, a dynamic protein that does catalysis at low temperature, ideally. And indeed, some of the highly stable proteins, they do not have activity at room temperatures, and some of them only start with their incipient activity at, let's say, 40, 50 degrees. And of course, this is something that is difficult to analyze for and difficult to utilize under desired conditions. However, there are many proteins, and this is astonishing, that have a quite rigid overall scaffold, but have a quite mobile residues in the active site, and therefore they do have or can have very high activity even at room temperatures, although the protein scaffold is rigid. And those are the interesting candidates, and of course you don't know that just from focusing on one or the other type. You have to investigate proteins that are thermostable and then look for activity at room temperature as well.
Okay, thank you. Here is another question about thermostable enzymes. Can we also conclude that the high thermostable enzymes are more suitable for nDSF screen?
I'm sorry, could you please repeat the question? There was some noise in
Sorry,
the line.
Dr. Fessner. Yeah. Can we conclude that high thermostable enzymes are more suitable for nDSF screening?
Well, this is something that I would not conclude. The technology is suitable for proteins with all properties, no matter what kind of TM. Actually, it's easier to measure something that has a low TM, because then the time to wait for the data is rather short. No, I mean, the technology is appropriate for all proteins, as I've shown. The technology is not appropriate, I believe, for proteins that are extremely stable, and there are organisms out there that that will survive temperatures above 110 degrees, and of course this is the limit of I believe the instrument to measure for. We've measured, I believe, proteins with stability up to 104 degrees, but this is certainly close to the real limit because of the heating rate that can be applied.
Okay, great. We have time for just one more question. What is the minimum % purity of a protein with 4 to 6 tryptophans necessary for sensitive measurement in nDSF?
Well, if I may comment on that. Sorry, yes.
Okay.
You actually have your experimental experience to comment on that. From our side, it doesn't just depend on the number of tryptophans, but also on their position. So even for proteins with similar numbers of tryptophans, you can get different fluorescence intensities. And for that reason, I would just say go ahead and try it. But Dr. Fessner, if you want to comment again, because you had some data on purity and signal from that.
As I said, we have a standard procedure. We've started with CFE with different concentration of protein, and we've noticed that at least at least a content of let's say 30%, 40% of protein content in the mixture is necessary to get reliable data on that. Of course, you can also see peaks for other proteins, but this is not something that I would interpret as safe, and I would always recommend to purify the protein in those cases to get good readings.
Okay, great. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their email addresses are shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Dr. Kern and Dr. Fessner, as well as our webinar sponsor, NanoTemper. Thank you everyone, and goodbye.